Hi, this is Professor Tiffany Martin with Medical Microbiology, and we are working our way through Chapter 3. This video is going to talk about the outside structures on a prokaryotic cell. So, everything that's outside of the cell envelope, so the outside structures, okay? So, here is your diagram that you're dealing with. So, the structures we're going to look at are the glycocalyx, which is the outermost structure. We're also going to look at flagellums there and oh, I forgot what the other one is. Well, we'll see when we get there. Oh, Fimbria, which is not shown in this picture, but we'll describe it when we get there. Okay, so the first structure we're looking at is a glycocalyx. Plural for glycocalyx is glycocalyces. This is the outermost layer on a bacteria cell, but it is optional, meaning some bacteria have one, some do not. So if you have the glycocalyx, that bacteria will always have a glycocalyx, but if it doesn't, it doesn't use one. So it is an outermost layer. It is made of polysaccharides, polypeptides, a lot of things in there. The functions of the glycocalyx is to protect it from drying out. It's just another layer on the cell that functions for protection of the cell. Now, there are two types of glycocalyces, which is plural for glycocalyx, and I kind of drew that out on my little whiteboard here. So glycocalyces, there are two types. There is a slime layer glycocalyx and a capsule glycocalyx. They're a little different. Again, both of them made of polypeptides, polysaccharides. Both of them will protect them from drying out from desiccation. The capsule layer is smooth. And it has an additional function in that it can protect a cell from digestion from the immune system. So those phagocytic cells, like the macrophages and other phagocytic cells, whose job it is to get rid of foreign invaders, they have to come into that foreign invaders and gobble them up and eat them up. And if a bacteria, if a bacteria has this capsule glycocalyx, which is very smooth, it makes it very difficult for the phagocyte to grab a hold of it. So bacteria that do have a capsule is more likely to be a pathogenic bacteria. All bacteria doesn't cause disease. Pathogens are bacteria that cause disease. If that bacteria has a capsule, it is more likely to cause us a disease because it is harder for our immune system to get rid of it. One example of a bacteria that has a smooth capsule, glycocalyx, is S. pneumoniae. This is a bacteria that causes pneumonia, and again, it makes it hard for our cells to get rid of it. Okay, second type of glycocalyx is the slime layer. This one's put together a little differently, and it ends up being very sticky. The function for this one is adhesion. So bacteria that have this type of glycocalyx are more likely, once they get inside us, to stick. To each other, to things inside our bodies, and then they stay. And if they can stay, then they may, time passes, they will replicate, and they potentially can cause us a disease. One example in this category is S. mutans. S. mutans is a bacteria that's commonly found in our mouth. So bacteria, we're constantly putting stuff in our mouths, and bacteria gets in there. That's why they tell you don't touch your face. It's a good portal of entry, comes into your mouth, and then the S. mutans gets in your mouth, and your mouth will try to get rid of it. Saliva tries to get rid of it. That's why our mucous membranes shed. They will try to get rid of bacteria. But in our mouth, we have teeth. It's a hard surface, and that S. mutans uses that slime layer, glycocalyx, to stick to our teeth. Anybody falling asleep in the middle of the night, probably are up studying, right? And you fall asleep, you wake up the next morning, and your teeth are all filmy and sticky because you didn't brush your teeth the night before. That is a biofilm. That is bacteria that are using their glycocalyces to stick to one another and stick to your teeth from that. Okay, so that's an example of how things stick. If you do not brush your teeth, that S. mutans and other bacteria will kind of change a little bit, and they'll form that hard surface that becomes that plaque that you can't brush away with just just a toothbrush. You have to actually go to the dentist and have it scrape away. So the ability to stay in an environment is more likely to cause us a disease, more likely to become for a bacteria to become a pathogen. So both glycocalyces are considered virulence factors. And again, those are those traits you're looking for in your case study projects that help a bacteria to get in, stay in, avoid detection from the immune system from that. Okay, so back to the PowerPoint. I think we covered all of that, benefits to the bacteria, and you can kind of see some pictures of glycocalyces. All right, remember not all bacteria have them. If they don't have them, they're less likely to be a pathogen. If they do have them, it makes them more likely to be a pathogen. All right, what else do we need to talk about? Let's talk about a flagellum. Flagella, you know this before, eukaryotic cells have flagella. Interesting note, the eukaryotic flagellum and the prokaryotic flagellum are very different structures. They have the same purpose, they kind of look the same from the naked eye, but if you look to see how they're put together very differently. They're made up of completely different proteins. These are examples of structures that probably occurred separately that do the same function, but they do not have a common ancestor. So they're put together very differently. Prokaryotic flagella, again, not all bacteria have them, so it's still an optional structure. If they have it, they will either always have it or at least the ability to make one. Some bacteria have produce flagella at different points in their life, depending on their environment. They're made up of flagellin protein. They're made up of three areas. One is the body, and this is what goes and anchors the cell into the cell wall. This is your cell membrane, right? Phospholipid bilayer, you can tell that. This is the cell wall of a bacteria. This is the body part of it. The hook is the bendy part, and the long thing coming out is the filament. Now, don't tell me it's a tail. I know that's what it looks like, but it is not a tail, so learn proper vocabulary. This is my visual aid of the flagellum, and we have the basal body that goes into the cell. We have the hook portion, and then this is called the filament. That extends and is capable of movement, generates the movement. Now, this filament is not rigid, so it kind of looks more like my adapted bendy straw. Here again is my basal body, my hook, and then the filament extending from that. From there. Okay, so that is the flagella and how it is structured. Let's talk about its movement. Now, if I were to ask you how a flagella moves, I typically get students that tell me this, kind of a wave light, which is true for eukaryotic cells. Prokaryotic cells, the flagellum moves differently. It actually rotates. So if I go back to my bendy straw and I use it through my hands, you get this rotation from that, this rotation, and that is how it moves versus that wave undulating motion that is characteristic of a eukaryotic flagellum. If I did this with my little filament that's not there, you can kind of get this movement. You can kind of see how it does that kind of movement. Anyway, so it is a rotation, it is a propeller. It can rotate that flagellum completely 360, and the bacteria can change the direction. It can go from a clockwise rotation to a counterclockwise rotation. So that's pretty interesting. And it actually ends up contributing to how this flagellum is going to cause the bacteria cell to move. If it moves in a clockwise direction, it's going to tumble. If it moves in a counterclockwise direction, it is going to run. Now, just a reminder, the scientific word for movement is taxis. So we can talk about movement being taxis for that. So clockwise tumbles, I don't know how you remember things. I think of tumbling, I think of a forward roll. So I think of clockwise going forward. So tumble is a forward kind of somersault. Counterclockwise, it will do the runs. So what's the difference between a tumble and a run? Well, when we look at a bacteria cell moving, here's my bacteria. If it is going on a run, the net movement is in a straight line. So that is a run, that is moving the flagellum in a counterclockwise direction, and the outcome is kind of a straight line. It's a run. When it switches and starts rotating clockwise, tumbling forward, roll forward clockwise, the movement becomes circular in motion, and you get this kind of pathway. And it will alternate runs and tumbles by changing that propeller. Okay, so why does it do that? observations for bacteria, we'll see that alternating runs and tumbles. It's not trying to go anywhere. You see, it just kind of alternates between runs and tumbles. If there is a food source, and it would like to go towards those nutrients, to those chemicals, it can move in a way that we would describe as positive chemotaxis. Food, nutrients, these are chemicals that it wants to get to. So we call it chemotaxis because it's moving towards chemicals. We're going to say positive because it is going to move positively in that direction. Now, if it was some negative thing, like you sprayed it with Lysol, you might have negative chemotaxis where it's moving away from that. So this happens. All right, now if it was you and me, we would just run to the buffet and go in a straight line from where I am to where the food is. But that's not what we observe in bacteria. They have to balance that with their desire to get to the food with energy output. And so what we see is still alternating runs and tumbles, but the net outcome is towards this food source. So again, it's not necessarily a straight line, but they still alternate runs and tumbles, but the net outcome will be towards that food source. When it's moving towards something or away from something, we see longer runs and shorter periods of time where they're tumbling. So they will do that more frequently. All right, you can also use these words with positive and negative phototaxis. What do you think they're going to? Towards light or away from light? And so that explains how that works. Okay, what else do we need to know about flagellum? We need to know how they're arranged. Typically, we kind of think of flagellum, we're thinking this monotrichous arrangement, where you have one flagellum coming out the end, but there are other arrangements that we see. There is an arrangement called a paratrichous flagellum. This is where you have bacteria all around, perimeter all around, covers the entire surface. There's another version called lophotrichous, where you have multiple flagella coming out of the tuft of a polar end of this. Lopho means tuft. I kind of think of lopho as a Dr. Seuss word, so I kind of remember this as lopho, like those truffle-up trees, so that's how I remember this one. Paratrichous, perimeter, monotrichous, one. There's another one we don't see as much. There's a bacteria shape called a spirillum that has this kind of corkscrew type shape. And when they have a flagellum, it actually runs along the corkscrew. The corkscrew, I'm saying shoe. It's corkscrew type shape. This axial filament type flagellum runs all the way along there. So again, that's very different than what you think of as a flagellum, but its purpose is movement. It works the same thing. Okay, I'm going to link this video into yours so you can see how that movement looks. All right, last outside structure. This is also optional. Not all bacteria have them. The ones that have them more likely to be pathogenic. This structure is called Fimbria, and there's these short little bristles that extend from the outside of some bacteria. They're used for adhesion. These are the flagellum, and you can see the Fimbriae are much shorter. Hey, what's your arrangement of these flagellum? I hope you said paratrichous. And paratrichous flagellum, these are the Fimbriae. They're much shorter. They're sticky. They're made of proteins. Their role is adhesion. Now, this is going to remind you of a similar structure for eukaryotic cells. Cilia. Yes, they are not cilia. Don't tell me they're cilia. They have a completely different function. Cilia are for movement. These are not. These are used for adhesion. It only looks like cilia because when we draw it out, there are these, extensions like that. So cilia, only eukaryotic cells have cilia. Some bacteria cells have these Fimbriae. Okay. All right. So sticky. We've shown how that helps a lot. A specialized form of Fimbriae is called a pili. This is a projection that can link two bacteria cells, and these are pretty cool. We're going to learn more about this in the DNA chapter. They also kind of referred to as conjugation pili, longer than Fimbriae, shorter than flagellum, and they will attach two cells for a short amount of time. The cool thing about this is bacteria will use these pili to share DNA. They can take pieces of their DNA and share it with another bacteria cell, which is really cool. We can't do that. You know, we pass our DNA down to our children, but we can't pass our DNA to our neighbor. Beneficial for bacteria because they can share things like antibiotic resistance, the ability to protect themselves from antibiotics. This is how we get those superbugs. These bacteria end up in the same environment. One of them is resistant to bacteria, its neighbor is not. They can form a pili between the two, share that information, and now both of these bacteria are resistant to antibiotics. So this is a big advantage for bacteria, and we'll talk about more again when we get to the DNA chapter. So what you need to know, it is a specialized form of Fimbria, and it mediates transfer of DNA. Okay, we're going to end that video there. Those are all the outside structures, and we will pick up with a bacteria cell wall.